It's my pleasure today to introduce Dr. Brittany Harlow. Hopefully, you all have seen Brittany before, and she's been with the Pasture unit for 6 years now. Brittany got her bachelor's in animal science and a minor in chemistry at the University of Illinois. Are you from Illinois?
Yeah.
Okay, from Illinois originally. She then got her master's degree in animal science with a specialization in GI microbiology and equine nutrition with Lawrence here at UK. Went straight from that to get her PhD, also with Lawrence, but this time adding Michael Flight with the on the committee there. And when she finished her PhD, she came on board with the faculty unit as a postdoc and worked with Glenn Aiken and then Michael for about 4 years. And she's been a scientist with them for 2 years. And she's soon to become a line advisor. I mean, everything goes quickly in the next year. So it's Dr. Harlow's research areas now include specific toxicosis, plant-based antimicrobials in livestock raising systems, and early lamb and calf microbial development. And I will let Brittany take it away.
Well, some of you guys are familiar faces and some are not. So I'm Brittany Harlow. I'm a research animal scientist with the Forage Animal Production Research Unit. Probably most of you have heard of us. But today I'm excited to talk to you guys a little bit about some of the research that we're doing. We're not going to talk about lambs and calves today, but we are going to talk about plant secondary metabolites and some of the work that we're doing with pasture nutrients. Yeah. All right, so I wanted to start out and talk a little bit about the forage animal microbial nexus, because this is really where my research program focuses, and a lot of our research that I'll be talking about today really centers on. And so essentially in raising livestock, all three of these components are both necessary and interconnected. So when we think about any one factor or changing any one factor, it's going to impact the other factors in this system. And so when we think about ways to actually optimize this nexus, we have to consider our goals for every component. And so different goals, right, depending on what we're looking at. So for animals, we might be interested in improving growth performance or health, reproductive performance, lactation, or even meat quality characteristics. With our forages, we might be looking to improve yield, or quality, composition, production of secondary metabolites, which we're going to talk about today, stand health, soil health, nutrient retention, the list is endless. And then for our microbial community that kind of intersects these two components, we might be looking at ways to optimize nutrient utilization or capacity and efficiencies like nitrogen and carbon. So methane, for instance, reducing the production of methane. Or detoxification. So the rumen microbial community plays a really important role in detoxifying compounds in the diet. Competitive exclusion of pathogens, barrier function in the environment. And then overarching goals, right? In the environment, how do these things impact the environment, like greenhouse gas emissions, contamination of water sources, erosion, ecosystem maintenance, and things like that. And so we have to think of all of these things together to really address optimizing grazing ruminants and animals. So what makes ruminants special? So a lot of you guys probably have seen this slide with my little animation, but I really like it, so I keep it for every talk. And essentially what makes a ruminant different from, say, a horse is that their first site of digestion is actually fermentation in the rumen. So when the animal consumes grass, it goes into the rumen where it's fermented by this metabolically and phylogenetically diverse group of microorganisms. And there's microorganisms in that group that can ferment almost everything that's present in the diet. And so these microorganisms take the pasture and its suite of nutrients, convert it into acids that the animal then absorbs to meet its energy requirements. And in grazing animals, as much as 80% of their energy requirements can be met by just rumen fermentation alone. And so it's a really important site or potential for increasing our efficiency of our livestock production on pastures, or even sources of inefficiency for production. So there are a lot of different strategies that we can use to optimize this microbial community in the rumen, and some of them we're going to talk about today. So The use of antibiotics and antimicrobials or antimicrobial growth promoters are commonly used to improve microbial efficiencies in the rumen. There's lots of different dietary supplements that are used in ruminant production systems. You might see on your feed label probiotics or prebiotics, or even microbial transfers can occur to improve rumen fermentation. Plant secondary metabolites, that's going to be a big topic for today, can also change rumen fermentation. And then, of course, just basic nutrient composition and structure. And that's where we're going to start today, because this has been something that our unit scientists have been working hard on thinking about. So this is the basic forage analysis for a pasture sample taken at the sheep unit. And as you can see, and all of you are very familiar with, there's these broad nutrient classes that we see outlined. We see crude protein, ADF, NDF, calculated values like NFC and TDN, and that's extremely important for our producers. They can use that to say, okay, here's the value of my hay crop, or here's the value of my pasture that the animal is moving to. But as scientists, and when we think about optimizing grazing animal performance, we have to think a little bit deeper than this, right? Because all of these classes are full of different nutrients with different characteristics, different concentrations, and we have to consider that when we think about optimizing our rumen function and animal performance. So one such example are fructans, and so fructans are the principal storage carbohydrate in cool season grasses, and our plant chemist, Dr. Isabel Kagan, is very, very good at studying fructans, and so I wanted to use this as an example of one type of nutrient that's present in cool season grasses pastures that dramatically can impact microbial fermentation. And so this here are two HVAC profiles of two different cool season grasses. Timothy here on the top, and then orchard grass on the bottom. And if you look at the chromatograms, you can see that she's denoted short chain fructans and long chain fructans, and also max DP, or degree of polymerization. And so you can see, just looking at these two grasses, there's a pretty vast difference in their profiles here. We see that the Timothy profile has more long chain fructans with a greater maximum DP, while the orchard grass has shorter chain fructans with a lower maximum DP. And so what does that matter, right? How does that really impact rumen fermentation? So then we looked at this in rumen bacteria. So there's Streptococcus bovis here. It's a resident fructanolytic organism in the rumen, which ferments these fructans commonly. And so it's an important bacterium in that process. And when we fed Streptococcus bovis either short chain or long chain inulin, short chain being the open bar and long chain being the dark bar, we saw that short chain inulin was more rapidly fermented and elicited a greater growth rate from Streptococcus bovis. It preferred, even at the same concentration, a shorter chain inulin. And that's also true when we look at a physiological response, or something that could elicit a physiological response like pH decline. We see greater pH decline with short chain inulin. So even something as simple as just structure can impact fermentation independent of concentration. And so to me, I really think that this is the low-hanging fruit of a way that we can look at our pastures and look at the nutrients in our diet and be able to optimize fermentation and productivity of animals. And so this research is continuing, obviously, with Dr. Isabel Kagan and Dr. Rachel Schendel, who's an arabinoxylin expert in the Animal and Food Sciences department. And they're going to be looking at variables that impact fructan and arabinoxylin concentrations and profiles of cool season grasses. And hopefully from that, we'll be able to look at species, season, environment, and how those all impact those profiles. And then finally look to determine how those can affect rumen fermentation and efficiency. And so this is all in the works, and stay tuned for more on this in the future. So the other half of this talk is going to be focused on some of our work looking at plant secondary metabolites and how those can be utilized to optimize rumen fermentation and animal efficiencies. We'll start by introducing the concept of functional foods. And so functional foods, you guys have probably all heard of them. Food as medicine, superfoods, nutraceuticals are a big concept in human nutrition because these foods infer health benefits beyond basic nutrition, because they contain bioactive food components or compounds that do so. And so for an example, is fatty fish. Nutritionists suggest that we eat 8 ounces of fish a week. fatty fish, and salmon because of the omega-3 composition. And so this is a functional food, and that just doing that can reduce your risk of cardiovascular disease, reduce blood pressure, and also reduce systemic inflammation in the body. And so these huge physiological benefits from eating a tiny amount of fish. And so that's what functional foods are. So how do we translate that to animal nutrition and health? Functional feeds. So we look at feeds that contain bioactive compounds and confer animal production or health benefits that when consumed regularly as part of the ration in a relatively small proportion can give those benefits. So they're really distinct from a feedstuff that just provides basic nutrition, things that can confer a benefit at a small level that doesn't require a great deal of nutrition in that portion. So there's lots of different compounds or plants that have been used as functional feeds or functional compounds from plants in livestock production systems. You've probably maybe heard Dr. Fleig talk a little bit about hops in previous seminars. That's something that we've looked into quite a bit for their beta acid concentration. There's also different oils found in cinnamon and garlic that can be utilized to optimize rumen fermentation in different scenarios. Thymol from thyme is a pretty common antimicrobial modifier. And then of course capsaicin that's been shown to, alleviate vasoconstriction and cause vasodilation. So all of these different plants have functional characteristics. And of course here I've included hemp because as you know, we're going to be doing lots and lots of research with hemp in the future. So stay tuned for lots on how this could be used as a functional feed in ruminant systems. So what about functional forages though? We're here talking about grazing animals. We're not talking about supplements and minerals and all that. We're talking about how can we look at our functional forages. So this is a picture of a mixed pasture. You can see that there are tons of different species of plants, great diversity, just like we talked about in the microbial community. There's all kinds of things here, a suite of phytochemicals that we can unleash to best produce the highest performing animal to confer disease resistance. There's this whole potential here that we still need to unleash to be able to do that. So there's been quite a bit of research done on this, and a lot of it has really been centered on tannin-containing forages in our pastures. So for instance, Sericea lespedeza, bird's-foot trefoil, and then here we have our forage chicory, and then even black locust trees and silvopasture systems have been used to achieve those benefits. And so tannins can exert potential benefits like antiparasitic effects. They increase rumen bypass protein, which is beneficial. We'll talk about a little bit later. And they also can bind toxins like alkaloids found in Enophytobacterium fescue. So in addition to that, clovers also contain bioactive compounds. And you guys have probably heard us talk a lot about clovers, but I'm going to talk about it again today because I think it's very important. And so why do we use clovers in our grazing systems? Well, they reduce nitrogen inputs, they improve pasture quality because they contain a lot of protein and are highly digestible. They improve forage diversity and therefore improve animal performance. And that's been repeatedly shown over the years. So is it really just about that digestibility? No, actually, red clovers contain large amounts of bioactive isoflavones, specifically biocannin A. And so these compounds are part of the flavonoid family and can act as antioxidants, phytoestrogens, and also have two newly identified benefits: growth promotion via biocannin A specifically, and then also vasorelaxant properties, which make it to have great utility in fast food toxicosis. So when we talk about rumen modification and that growth promotion, what we're really looking at is red clover, those isoflavones as antimicrobial growth promoters. And so what is that? An antimicrobial growth promoter is an antibiotic or other antibacterial agent that selectively kills certain bacteria in the rumen, not all of them, certain wasteful bacteria, to alter how nutrients are utilized and allow the animal to gain more weight and improve their feed efficiency. And so we've seen products like Bovatech and rumensin are commonly used in cattle production systems for this exact reason. So another way to look at rumen growth promoters, antimicrobial growth promoters, this is Michael Fleist's example of how he explains it, and I think it's a very good way to do so. So in our pastures, we have forage species and weed species. the forage species we want to propagate, those good bacteria we want to propagate in our rumen, but we want to kill off these weed species. And so we use herbicides to do that. In the rumen, we use antimicrobial growth promoters. So again, in the rumen, we have beneficial species like those that utilize forage or fiber. And then we have wasteful species like these methanogens here that are wasting carbon. And we want to kill these and we want to keep these. And so that's exactly how those work. So most antimicrobial growth promoters really target protein fermentation. And so when protein enters in the diet, hyperammonia producing bacteria in the rumen will ferment that protein or its constituents, amino acids and peptides, and produce an excess amount of ammonia. And so only a little bit of ammonia is required by the ruminal community to meet its requirements. So a lot of it is actually excreted out in the urine and wasted. So we lose a lot of nitrogen that way. And so where antimicrobial growth promoters like monensin come in is that they inhibit these HAB and therefore stop that wasting of ammonia and increase bypass protein. So the protein that makes it to the animal to be enzymatically digested and can be used to make milk and meat. So another aspect of rumen fermentation that's extremely important, arguably the most important part, is that the enzymes that break down fiber are strictly microbial. And when we have a pasture-based diet with lots of fiber, this function is essential to our animals' productivity. Unfortunately, some antimicrobial growth promoters actually inhibit fiber fermenting bacteria. So, for instance, monensin actually inhibits fiber fermentation. So it has benefits by killing off protein utilizers, those HABs, but it has negatives by killing off fiber utilizers. So here's kind of a compilation of several studies, 7 Pasture studies, where they looked at the efficacy of either Bovatek or rumensin in grazing systems. And what they found were variable results in different studies. Sometimes we see great growth promoting benefit, sometimes we see actual reduction, sometimes we see no effect. And likely that is related to that fiber inhibition and the specific systems, making antimicrobial growth promoters like monensin to have decreased utility in grazing systems, or at least you'd be a little bit more cautious to utilize these growth promoters. In contrast, biocanin A from red clover is a selective inhibitor that actually inhibits protein and amino acid wasteful fermentation in the rumen and improves fiber utilization, which likely results in increased short-chain fatty acids or acidic end product production that the animal absorbs to meet its energy requirements and likely translates to increased growth. And so we've done a series of studies looking into that in the field that I'm going to cover a little bit today. So the first study was done by Dr. Glenn and Michael, and I happened to come in for my postdoc right at the tail end of this research, where they supplemented purified biocanin A to grazing steers on non-toxic cool season grass pastures. And so they had a pasture-only treatment, they had a dry distiller's grain supplemented treatment, and a dry distiller's grain plus purified biocanin A, as much as fits in your hand, maybe less than that. And what they saw was that biocanin A alone improved average daily gain significantly. And so a very large response just from a purified isoflavone. So yes, it works, but it's way too expensive, right? So purified biocanin A, we can't afford to put that into cattle rations. We gotta, work by our bottom line here. So then we were moved on to look at some other supplementation possible strategies and levels. And so in this next study that we conducted, we looked at 2 levels of red clover hay, 15 and 30%. And so the high red clover level was 30%, which was made to be similar to that previous study by Dr. Glenn. And then the low red clover level was half of that, so 15% of the diet. And then we had a DDG matched protein control. So all of these had the same amount of protein included in the diet from different sources, of course, but 2 different levels of red clover. So what we found was that the low red clover treatment actually produced the greatest growth benefit. And so that, serves to that functional feed or forage hypothesis, lower levels, greater promotion. And we were able to recapitulate the same results that we saw in the lab. We saw reduced HABs with red clover. We saw no impact on those cellulitic bacteria or fiber utilizers, and saw a great improvement in hay digestibility in those rumens. Because see, we grazed a fistulated Holstein here to serve as our microbial donor, so we could look at that. And again, the low level was most effective. So through this process, we talked about small ruminants. What about small ruminants? And so we worked with Dr. Donnie Lee at the sheep unit to look into some of the applications, and this is some newer research. And so most recently, my current postdoc, Dr. Jen Weinert-Nelson, did this project, and she took ram lambs, and all treatments received 85% concentrate, so it was a high concentrate diet with 15% forage component. And the concentrate consisted of corn and dried distillers grains. And so for the roughage treatments, what we saw was 15% orchard grass, control, 7.5% red clover, and 7.5% orchard grass, so half and half, and then a 15% red clover treatment. And they had ad libitum access to this diet. And in result, we saw great improvements by just switching out that forage component with red clover. And it's important to note that these diets are balanced for energy and for protein. And what we saw for average daily gain, we saw that both the 7.5 and 15% red clover treatment improved average daily gain by as much as 15%. And we saw a 22% increase in a gain to feed ratio. And so essentially, what I mean by gain to feed is how much gain per unit feed. So higher is better, more gain per unit feed. And this is pretty substantial when you think about the economic bottom line of these type of finishing systems. So based on this, we have a lot of conversations, red clover versus alfalfa, right? And then when we talk about isoflavones, at least, they're differences. They have great differences in isoflavones. Red clover containing so much biotin A. And so when we think about these different legumes and their application in diet in this way, alfalfa certainly is still the queen of forages and is a very marketable feedstuff, but red clover may actually be a functional forage in that it confers additional benefits. So what's next with this? So we do have 5 years of research plan with Dr. Ely to expand on this research in small ruminant production systems. And that will include routes and supplementation. So we will be getting out to the pasture, looking at overseeding and hay, the possibility of utilizing a pelleted red clover supplement and also mineral. We'll be looking for the optimal dose and different finishing systems for these sheep. And then also carcass characteristics, which is a new area for us at Pasture to get into with Dr. Suman. And then finally, reproductive effects. So in sheep and sheep production, saying anything about putting red clover in the diet is a big no-no. It means infertility. They don't want to give it to their ewes because it causes at high levels infertility issues. And so we'll be looking a little bit more into that, looking at the levels that are actually applicable for growth promotion to see if those have any fertility ramifications. So another aspect of what we're looking at related to this project are some forage variety trials. And so our next project plan, Dr. Ray Smith has been helping with us to start now, and Gene Olson. And what we're going to be looking at are 3-year evaluation of legume variety trials. We have collaborators in Wisconsin and Alabama that will also be submitting samples for this. This is what Elena Jacobs will be working on when she finishes her PhD. And we'll be looking at factors like species and variety, plant characteristics like maturity, plant part, and also trichomes, which you've probably heard about in her 3-minute thesis talk last week. And then also season and environment. And so stay tuned for some of that as well. So what happens to biotin A and the other isoflavones after they leave the rumen? So they're metabolized by those rumen bacteria and changed to make them more soluble. And then the metabolites enter the blood where they can have several different impacts, including decreasing hypertension or acting as a vasorelaxant, which gives them some utility in toxicosis or heat stress. So for a long time, many years, we've known that cattle that are grazing toxic tall fescue do better when there's clover in the pasture. And a lot of that has really been attributed to better nutrition or to dilution of the alkaloids in the diet. The animals are consuming less toxic alkaloids because they are eating another forage or a higher quality forage. But is it really dilution or is it vasorelaxation? So you guys all are familiar with fescue toxicosis, but we'll go through my little figure here. And so when an animal consumes toxic tall fescue, it causes peripheral vasoconstriction, leading to several detrimental effects that can occur. And so things like decreased prolactin is kind of a hallmark of fescue toxicosis. We see a lot of winter coat retention or regrowth in those cattle. They're hairy, shaggy, And then vasoconstriction specifically impacts things like heat dissipation. They have lower heat tolerance, absorption of nutrients, and then of course reproduction. So Pasture has been working on toxicosis for years and years and years. Dr. Jimmy Koff is in the crowd. He's an expert on toxicosis. Dr. Glenn Aiken, who I did my postdoc with, did a lot of field work on toxicosis before I even got here or before I even finished my PhD. And so I wanted to tip my hat to some of that research that had already happened. So Glenn had done a lot of work with his technician and now my technician, Tracy Hamilton, looking at soy hull supplementation, the application of chaparral to reduce seed head emergence, utilizing novel endophytes and things like that in grazing systems. And he had a very successful career looking at all of these things. And actually, towards the end, him and Dr. Blythe had the idea that red clover might have these vasorelaxing properties and might influence toxicosis. And they did a little goat experiment where they showed that when they used a menopausal supplement that contained red clover, they were able to alleviate vasoconstriction and prevent vasoconstriction in goats challenged with hot tall fescue. And so that was kind of the basis of where the next body of research I'm going to talk about came from. So first, I want to explain how we look at vasoconstriction and vasodilation. And so Glenn Aiken, this was one of his hallmark measurements. He used color Doppler ultrasonography to look at caudal or carotid areas, depending on what species you're looking at. And he could actually see vasoconstriction and vasorelaxation. And so I like to use this analogy to talk about this topic. So when you're drinking a milkshake, do you want to use a coffee stir or do you want to use a milkshake straw? Anybody want to use a coffee stir when they drink a milkshake? I don't. I want like 2 of these straws, right? And so when an animal has fescue toxicosis or suffering from fescue toxicosis, their artery looks more like this than this. And so you can imagine the increased pressure, the reduced blood flow to those peripheral tissues, and how that could have some really severe physiological ramifications for the animal. So essentially, the alkaloids of toxic tall fescue are vasoconstrictors, and the isoflavone metabolites in legumes like red clover are vasorelaxants. So we've looked at several isoflavone supplementation strategies. As I mentioned, Dr. Fleith and Dr. Aiken started with a woman's menopausal supplement that was supposed to alleviate hot flashes, but we can use things like interseeding red clover to achieve the same benefit. And we did see that when animals were grazing either toxic tall fescue with no clover that was fertilized or interseeded red clover, they saw an improvement in vasodilation and a very large improvement in average daily gain. And so again, recapitulating this concept that red clover improves tolerance to fescue toxicosis. We've also been able to use red clover hay supplementation on pasture. Part of the reason for doing this was because it's a little difficult to establish 15% red clover pastures, as I'm sure many of you guys know. And so we looked at 15 and 30% levels in steers grazing identified affected tall fescue pastures and balanced those diets for protein with dry distillers grains. And what we saw was that both levels improved vasodilation and both improved average daily gain, a similar amount as what Glenn observed in his previous experiment. And then one of the big questions we were getting from producers was, what about other isoflavone sources? I have soybean meal in my diet. I have white clover already in my pastures. Why do I need red clover, right? So, well, these other sources work the same way. What we found is that weight to weight added into the diet, all of these sources improve vasodilation with fescue challenge in fistulated goats. So this was really interesting because if you think about these different sources, they all vary in concentration and composition. So for example, white clover has 25 times less isoflavones than what we see in red clover on average. And soybean meal has a flip-flop profile of isoflavones, very different than what we see in red clover and white clover. And so this led us to believe maybe we can go a lot lower, Why are we thinking about feeding levels? Maybe we need to think about, functional forage levels, right? Very low levels. So we went for it. We met with Bergman Nutrition and we came up with a project to look at including brown red clover leaf, which Tracy Hamilton loves to prepare by shaking red clover bales, collecting the leaf and then grinding it through a sieve and then mixing it in conventional loose mineral. And so we had 4 treatments in this study. We had a 0% treatment with no red clover, a 10, a 15, and a 20% treatment weight to weight red clover. And so this kind of looks like feed here, right? You see a lot of green leafy tissue. They did not consume it like feed. We had done a study prior to this looking at palatability and consumption rates with this clover versus the conventional, and saw no difference. So the salt in there still did its job. And what we saw was that when animals received the conventional mineral, we saw vasoconstriction with fescue challenge. But with any of the minerals, we saw an improvement in that. All of them were effective at mitigating that vasoconstriction. So as much as a handful of red clover leaf was enough to make an animal not suffer from vasoconstriction, from a pretty substantial fescue challenge in the pens at least. So then we decided we need to take it to the field. And so again, back to the hypothesis that there might be some reproduction ramifications of including these legumes in diets, these high phytoestrogen containing legumes. And so we went ahead and did this project with beef heifers, looking at fertility as well as part of the project. And so this study was conducted in spring 21, and we just finished it up in October this year. So some of that data isn't in here, but I will share with you what we have. And so we had beef heifers that were grazing endophyte infected tall fescue pastures, and they had one of two treatments, either a mineral control treatment, so that conventional mineral treatment, or a mineral plus 20% red clover leaf. And this was done in collaboration again with Perfect Nutrition, Eden Shell Farm and Kentucky Beef Network, and then Les Anderson from University of Kentucky. And what we found from year 1 and 2 was that there were really no differences in mineral intake, body weight, or body condition between the treatments. All of the animals did relatively well, given their circumstances. And we saw really great fertility rates regardless of treatment. It had no issues related to that, and suspect that there are no issues. Although conception rate data requires a great deal of number of animals, so we certainly will be continuing to make sure that this is correct. So some of the other variables we looked at were things like hair coat score. And so we use this hair coat scoring system from 1 slick short summer coat to 5 full winter coat. And so in fescue toxicosis animals, as I mentioned before, we see a lot of extra hairy cattle out on pastures when they shouldn't be hairy because of that fescue toxicosis. And what we saw was, this is just year 1 data. Then on 4/21, which is the first day of the experiment, we saw that the control treatment had a lower hair coat score than the red clover treatment on average. And so there were some blocking issues in year 1. But over time, that flip-flopped by 7/16, that the red clover treatment had a lower hair coat score than what we observed in the control. And so overall, there was a pretty big difference between those treatments on hair coat score change over time. So another variable that we looked at related to heat stress was respiration rate. And so we measured those serially on pasture every 2 weeks, and we recorded the temperature and humidity at the time of that measurement. What we found was that, again, the red clover treatment had similar respiration rates at the beginning of the study, but as it got hotter out, we saw that the red clover treatment had lower respiration rates than what we observed in the control treatment, indicating that red clover actually was able to help alleviate some heat stress in these animals. And this was matched with improved caudal artery areas in those animals. And so we did see vasodilation and a relieving of heat stress with red clover mineral. So again, back to the straws, with red clover, we saw that they were opened back up to milkshake straws. So what's next for our isoflavons in fescue toxicosis? So we still have to determine an optimum source and concentration for these isoflavones and their role in this. Of course, we've gone pretty low. In the heifer project, we used 20% weight to weight or red clover in those treatments. But we also saw in the pens that 10% was effective as well. And so how low can we go, and what compound do we really need to see the best benefit? Potential different routes of supplementation. There's been some work being done on potential ear implants, or possibly lick tubs could be an option for supplementing these isoflavones at low levels. And then other potential applications. We see this alleviation of heat stress, dairy systems. There's other applications that require that would benefit from that utility, and I think could be researched further. So the last thing I wanted to plug today is a little unrelated to the research that I've been presenting to you, but is a big project coming up for our laboratory. And what we're doing is we're doing a Montana-Kentucky heifer swap project. And so we're taking NFI naive heifers from Montana and bringing them to Kentucky, and we're raising them either on hot tall fescue or on NFI free tall fescue to see how they respond to that. We'll be looking at things like microbial community establishment, the animal's health, as well as their reproductive function. So the idea and the goal of the project is to profile heifer and calf physiological and rumen microbial adaptation to different raising environments, as we'll also be sending some cattle to Montana. And then we also will be using this to identify some phenotypic and genetic variation of cattle for tolerance of fescue toxicosis. And so this project is a little bit off the beaten path of what I've been talking about today, but certainly presents an interesting concept of project that will be coming up soon. So look forward to those results. The last thing I wanted to do was thank the Fabry research team, specifically Tracy Hamilton, my current postdoc, Jen Weiner Nelson, our interns and the other Fabry scientists and technicians for their contribution to this research. I tried to give everyone credit where credit was due. And of course, all of our collaborators with University of Kentucky, other universities, industry, and our stakeholder collaborators. So with that, I'll take any questions. I've included my contact information on the slide, so if anybody doesn't feel like asking me a question right now, you can send me an email or give me a call. I'm happy to talk about this anytime.
Great, thank you, Brittany. Have we got any questions?
Brittany, I know Berkman Feed has said that they would like a refined biotin A to be in a mix as a supplement, not red clover leaves. And obviously it would take some effort to get red clover leaves, but is that just because the trouble, or does the level go up and down that they don't think can be consistent?
I think they have concerns about the consistency of their mixing process with the red clover material and the mineral itself, and so they prefer to use a purified or an extracted compound instead.
If you could get the mixing down, do the levels stay within a, somewhat good range, or do they go up and down in the red clover leaves?
So we've had a lot of luck, all out of the same batch of hay, being able to replicate a similar mineral every time when we mix it, because we do hand mix our mineral at the farm to do that, and we've been pretty successful in small batches of doing so. I imagine though, like I mentioned with the variety trials that we're going to do, that batch to batch red clover is going to differ, and that may also impact the levels in that mineral. And so I think all of those considerations have to be made when we think about an actual product.
You'd almost have to measure each batch.
Yeah, some kind of quality control likely.
Are there that many red clover hay producers in the state?
There's probably no pure red clover producers, but a lot of producers that are growing grass hay would be including red clover. I guess that's the other thing, is that we need to be really growing pure red clover to have consistency. In Michigan, there are some people growing pure red clover.
Why is that? Why do they grow it there?
I guess in their dairy market, I mean, it would be the market that would buy it. And there's, I don't know, Brittany, if you were telling me, or I think it was a horse manager, that Claiborne Farms used to feed red clover hay, and they felt like they got some real health benefits by giving just a little bit of red clover to their horses, particularly on the track. And it's curious. I heard that years ago before you guys did this work, and you heard that, or any?
Tracy, is that where we bought our first hay batch from?
Yeah, they were feeding it to just like their stud horses.
Yeah.
What I don't remember, can't remember why.
I need to talk to this farm manager who was related that to me just a few weeks ago, and try to get a little bit more.
Yeah, but they
that
something like, well, I think they went through this is pretty simple. They just went through a few batches where it was kind of it was a little mature, and so they started having some trouble with the stimulus, kind of injuring their mouths a little bit. That's what the guy told us, but there was a reason why. I can't remember for the life of me why. He said that they had certain, like some stud horses that they fed. They like to feed the pure red clover to, and to add to the why Michigan. So the first couple of years of doing this research, that was where we found pure red clover hay was up north. And that guy that trucked it down there said they basically use it in like their row crop rotation. So they could get a good 2 years out of the red clover stand. This kind of part of their rotation is why what he explained to me is why they, had the pure red clover. And
So not a cover crop, but actually like a multi-year
Yeah.
perennial type system.
right.
If hay producers, you told them that they would be paid a healthy dollar.
Right, if you told producers that, yeah, we got a market for red clover hay, it's like every other ag commodity, everybody will grow it, so. Tell them they'll make a buck or two.
Yeah. So maybe improved male reproductive
Well, not-
health as well.
Yeah.
We might all make a few ideas.
I think we have enough defunct hemp processing operations out there that could do what we need to do with separating clover leaf and stem. I think those machines could do the same process.
Okay.
So, I mean, they're out there, and so I think if you get some producers to grow some acreage of red clover, and I've seen her give a couple of talks. One, there was a guy that owns a small mineral company. He said, hey, I'm ready to, he was ready to, he said, give me the clover and we'll make a mineral.
Any other questions? Online Zoomers, feel free to chat or speak up.
I have a question. This is Erin on Zoom. Can you hear me?
We can hear you, Erin.
Okay. I'm interested in that, like the stemminess issue, and I wonder if something like mowing could be used to try to reduce that, or some other type of management. And then also, like, and I might have missed this in Ray's question, if there are different varieties of clover, or like medium red versus red, that might have different profiles, and if you're exploring any of that with the research.
Yes, so Elena is going to have the joy of exploring that quite extensively, looking at different varieties and different sources, legume sources, and how those might impact those levels. Dr. Isabel Kagan's done some work looking at different red clover varieties and alfalfa for isoflavone concentrations as well. And so that's something we certainly are planning to explore further. And I'll defer the question about mowing to Ray or to Tracy if they would like to speak on that.
I think it would be pretty easy within a growing season.
Well, we were talking about hay, the hay, not grazing. I guess that's what she's asking, right? About. Is it?
Or whether you could just mow it and then pour hay, reduce the stemminess, like if mowing in general would make it just produce less stem.
Sure, yeah, I mean,
Lower.
getting it cut at a earlier maturity would certainly help.
Yeah,
Right.
well, then it could be your first cutting is gonna be stemmiest.
Yeah.
It often is harder to dry. But a second or third cutting, typically just from the growth standpoint. So you can probably take care of that just by the timeliness of cutting at the time of the year you cut.
So is anyone breeding clovers for isoflavone specific goals?
There's work in New Zealand to breed, what is the, birth control
Yeah.
compound?
From mental.
Yeah, so breeding to try to reduce the,
Equal,
yeah.
are you thinking of equal?
I think so. So the phytoestrogen compound.
Yeah, equal is what is, I guess, it's what's produced in the rumen. But it's formalin that's the precursor.
So they've worked to try to reduce the amounts of that, because obviously you don't want to inhibit reproduction. And in talking to some of the breeders and hearing about, you guys' work, they've said they feel pretty confident that they can shift the levels of that, and they can shift the levels of like in an egg. Do you know of anything further with that?
I don't know anything further about any projects like that. Do you, Dr. Fly?
I think, Jimmy, you have some recent results with Equal, don't you?
It does cause nasal relaxation more than the native compounds of the original. So it definitely seems like the metabolite has more of a direct effect anyway, is what we've looked at in the diagram.
Okay, so let me see if I'm getting this right. The animal is eating for metformin or whatever it's called, and the
We'll call it
plant,
Rupert.
okay? But then the rumen is converting it to equal, and equal is the thing we think it's doing the, okay, got it.
But that's different than biotin A.
Mhm.
I just commented on that because breeders have bred to reduce levels, so they can shift levels of that. And so the clover breeders I've talked to in New Zealand feel quite confident that they could probably similarly shift levels of biotin A if they really worked at it.
Yes, ma'am.
Do we know why red clover produces biocanin A? And if there's environmental variables we can exploit to make it produce more?
I feel like I'm just deferring all my questions that Isabel Kagan would have the best answer to that question.
Well, I'm not sure why it's produced. Well, with a lot of these secondary metabolites, we don't know exactly why, but there's thought that they provide some sort of an ecological advantage. Isoflavones, I think, are like flavonoids in providing some sunscreen protection. They're formininettin is a precursor to an isoflavone, to a clover phytoalexin, so an antimicrobial compound that may aid in protection against plant pathogens. I'm not sure of whether biocanin A has any such relationship to antimicrobial compounds. Biocanin A is a precursor of genocide, if I've got that right. And so there may be some, if there is some benefit in the clovers having these compounds present, they're going to stay around. It's like other secondary metabolites. We often don't know exactly why, but it's thought they provide some sort of a niche, some sort of an advantage to help the plant stay around. Plants can't run away, so they have to have some of these compounds to help them survive.
Tomo, did you have a question earlier?
Yes, I was just curious about seaweeds because I found the news talking about seaweeds as a potential animal feed and reduces methane production significantly. I'm not sure whether it's true and if it's like a great potential for it, or any thoughts about seaweeds?
To be honest, I certainly am not sure. I think there's been multiple results in literature in terms of potential versus it not being ineffective. It's likely related, just like these studies, to specific batches or specific circumstances. But for what I read, there are some opportunities there potentially, but I'm not entirely sure of the utility.
So no projection or in a real like industry level yet? in
Not
terms
that
of
I'm
nutrients?
aware of.
Okay.
No.
But there are companies that are putting seaweed in their mineral and making great claims.
Yeah, I think it's all anecdotal. I don't think there's been much science.
Yeah, Jason.
So I was just curious if the mechanism of vasodilation is known, is it like an ACE inhibitor or an ARB or something?
Well, again, Dr. Jimmy Kloss would probably be better to talk about this.
It stimulates nitric oxide.
Oh, okay.
That is the source of the dilation.
It stimulates nitric oxide.
Nitric,
Oh,
nitrous is good.
yeah. Well, any other questions, Zoom land or in the audience? All right, well, let's thank Brittany and Pasture